Hello, Eugene Aiello here. Welcome to my next video in the series. I hope that last video did not lose you, because that whole concept of semiconductors is going to be the nature of what we're going to discuss pretty much in the rest of this biophotonics. If you're having trouble with that, I would just suggest even listening to it in slow motion. In my text, the Conscious Canary, I really detailed it out pretty good. Even amino acids, where I gave you a general amino acid in my description, I get more specific in the book, explaining how the different amino acids act as extra electrons. The pi electrons act as those extra electrons in the semiconductor material, and how different amino acid residues would actually act as holes that they'd want to move those electrons over there. And charge dynamics, I kind of breezed through it. I talked about ATP, how ATP can change the charge dynamics and affect how these proteins become semiconductors. But also keep in mind, and we're going to discuss more in this topic here, that electromagnetic energy, biophotonic energy within the cell. So I'm not talking most of these protein interactions, it's not occurring from that solar electromagnetic energy. That interface is happening in the pigment system. Those keratinocytes, the keratinocytes that I discussed earlier, where they have those pigments around their nucleus, those cells are actually exposed to solar electromagnetic energy. So they're able to create their own system within the cell. The rest of the cells in the body are going to be dependent on this biophotonic system, where that pigment system is going to be transferring that energy into what I call the glial system, which we'll discuss later. But right now I'm going to go right back to a cell though. So this could be any cell of the body. It's getting electromagnetic energy either through the biophotonic system, or it's at the surface like a keratinocyte would be. So we need to discuss microtubules and basic cytoskeleton of a cell. Proteins take on many forms in cellular life. Enzymes we talked about. But what's very important is we have a system in every cell called the cytoskeleton, which is composed of a peripheral cytoskeleton, which has actin, myosin, intermediate filaments, different type of fibers that create an actual perimeter just underneath the cell wall. You also have interlinking. So this cytoskeleton, this peripheral, it's not visualized well, but these proteins are all throughout the cell. When we get to talking more about structuring of water, water structures around proteins, actually creating like a gel. So water is not in a liquid bulk water state in any cell. It's in a gel structure around these protein networks. So the big protein network is the microtubule network. The microtubule network is going to emit from an area called the centrosome. The centrosome is located right next to the nucleus of the cell. Within the centrosome, you have two structures called centrioles, which are composed of microtubules in groups of 9 triplets, meaning if a cross-section, I'm sorry, my artwork isn't that great. If you did a cross-section of a centriole, it's going to be composed of microtubules in groups of 3 that are interlinked, and there's 9 groups of those. So at the center of the cell, we have 2 centrioles, and they're in an area that's basically called interconnecting fibers. It's just a grouping of different proteins which are interlinking all throughout in a lattice. You have some microtubules that are going to project directly from this interconnecting fiber network. More importantly, you have 2 centrioles, one being called the mother, one being called the daughter. The mother centriole has appendages on it called subdistal and distal appendages. The daughter centriole does not. So over here I talk about how the mother centriole is more complex. It has subdistal and distal appendages. These appendages attach to microtubules. So microtubules projecting off a centriole are only going to occur from that mother centriole. Only during mitosis, cell division, does this daughter centriole then get modified to where it has subdistal and distal appendages. And it basically mimics, it's creating the same structure as that mother centriole as the cell divides, and now it has a pair of matching mother centrioles. Hopefully I didn't confuse you on that, but not a topic we're too worried about here. So these microtubule systems project all throughout the cell. Some of these microtubules will actually connect to that peripheral cytoskeleton and be directly in contact with protein receptors. So all along the cell are these transmembrane protein receptors, which are very important. When we get into conventional biology, they talk a lot about lock and key fitting. Different hormones will attach here, like insulin is the one I'm going to talk a lot about. Insulin molecules will lock in there. That will create an interaction which allows the cell to take in sugar. These microtubules have a plus end and a minus end. They're constantly growing. They can change their lengths. This could be used in many ways. This could be used to change the shape of the cell. So when we were talking about those dendritic cells, the Langerhans cells and the melanocytes, those finger-like projections are created by microtubules pushing that cell membrane out into a certain shape. So this plus end is constantly getting added to and subtracted to change the length of the microtubules. Microtubules are composed of tubulin subunits, which are made up of an alpha and a beta tubulin subunit. These are basically just protein coils, mixtures of those alpha helixes I discussed earlier and beta pleated sheets in series. The alpha subunit is negative, the beta tubulin is positive. So they stack on top of each other and they create a coil structure of those alpha and beta tubulin subunits lining up. I'm going to take an offshoot little mention here. I find it very interesting that when a lot of quantum biology theorists out there are talking about these subunits as being qubits of data. Justin Riddle, excellent podcast that he has. Not podcast, it's a YouTube channel. He delves way into the concept of quantum biology and quantum consciousness, how these guys are processing data, which we're going to get to in a little bit as I explain more. But keep in mind that these arrangements of these tubules create a pattern, a structure that electron flow and electromagnetic energy is going to interact with. So going back to this concept of that centrosome, in my biophotonic model, these centrioles are creating the dipole antenna, which is receiving electromagnetic energy. So our body has a field of electromagnetic energy, a coherent wave, which is made up of multiple frequencies. We're going to describe more of that later when I start talking about quantum physics, but very important to note that this area here in my model is the dipole antenna receiving all information from the rest of the body, from the main function of the organism. That quantum energy that's flowing through it that I talked about originates in those pigment systems. So that microtubule, that centriole, is made up of these 9 groupings of triplets of microtubules. I couldn't help but to think when I was figuring all this out, I was already clinically working on testing people with different frequency lasers on different nerve dermatomes. And I was finding that there was a consistency that certain nerves would stimulate with certain frequencies. Those frequencies I found were red, green, and blue. There was 3 different frequencies, and then there was another near-infrared frequency when I talk more about clinically how I figured this out, that also had an interaction. So there's 4 different frequencies, biofrequencies, that I believe that the system's working at. Every cell is absorbing different combinations of those 4. So when I was looking at this triplet configuration, it just stuck out at me right away, red, green, and blue. When you looked at the length of a centriole, centrioles are about 500 nanometers in length. So if you look at how a dipole antenna functions, a dipole antenna is a quarter of the wavelength. So the length of the active dipole is a quarter of the wavelength of what you're trying to receive. So what that means is the complete length of one of these centrioles is 500 nanometers, which equates to 2,000 nanometers, which is in the near-infrared spectrum. If you remember, I talked earlier that mother centriole has a distal and a subdistal appendage. So those appendages, if you come back over here, and you're looking at that right angle orientation of the centrioles, this mother centriole has distal and subdistal appendages. These subdistal appendages are going to be found attached to every one of these triplets. The distal is only going to be found on one of those 3 triplets. So if we come back over here, I'll try and take this slower, I know I'm going quick. We've got a distal attachment site on one of the 3 centrioles, rods, within one of the triplets. And then you got a subdistal appendage attached at some location on each one of those 3. what I figured out is if you go to 187 to 153 nanometers from the bottom here, this bottom here would be where the interconnecting fibers are. So those interconnecting fibers would be the connection of the dipole, the daughter being the ground. So if you connect this up from 187 to 153, 4 times that, you're going to be at 748 nanometers to 612 nanometers. That would be within the red spectrum of light absorption. If you go to the next one, so that would be a red frequency pickup. The next one, 142 to 123 nanometers times 4, puts us at 568 to 492 nanometers. That's been within the green spectrum, 570 to 495. The third of the triplets, we could be at 123 to 112, which would be 492 to 448 nanometers, putting us with absorption of the blue spectrum. So I'm theorizing that mother centriole, those triplets are configured in a certain way to pick up the frequencies of red, green, and blue light, and then one of those 4 is going to be picking up near-infrared frequencies. What gets important is how does this dipole arrange? Meaning, are each one of these triplets connected to a triplet and a daughter? When I get into stem cells and how stem cell differentiation, which will be a whole video, I think that stem cell differentiation is just a matter of how these different groups are connected, how that ground is set up. How the variations of those things are set up depends on how the cell is going to receive those frequencies. So what does this mean? When these frequencies are coming into this system, it's going to be putting these frequencies into this microtubule processing system. What Justin Riddle and many others have said is a quantum processing system. This is able to process what's going on in the cells. So different interactions with proteins, which do exist on these microtubules, those could be interfaces of data into this quantum system. This quantum system is taking those combinations of frequencies that I just talked about there, making this a unique cell, which will process through there, and with those semiconductor natures of that protein network, those microtubules could be emitting light frequencies. Those light frequencies could be affecting how a mitochondria is going to produce ROS, how it's going to make energy. Those light frequencies could be affecting can be affecting genetic expression. There's microtubules that run from this interconnecting fiber network right into the nucleus. When a cell is dividing during mitosis and splitting, these microtubules are helping split that DNA and replicate it, and then migrate 2 different nucleuses to each different pole of the cell as it's dividing. So the interaction of these microtubule systems with the genetics is well understood. It just hasn't been put in place that there is an electromagnetic component. Conventional science, somehow they're saying that these microtubules are actually mechanically doing it. There's so many enzymes that are replicating the DNA and transferring everything around and organizing it. It has to be organized in a certain way. Those proteins are all absorbing electromagnetic energy and interacting and doing things based off of how that structure is set up in those cores of the proteins, the semiconductor nature of the protein. So this electromagnetic system can even govern how things move around. So there's proteins attached to these microtubules. You're familiar with one of them already because I discussed it. Those melanosomes that are produced in the melanocytes, they can dictate how these things move around in the cells. So if we produce certain products, like a salivary gland is producing saliva, the DNA is going to be told to produce that protein. Those proteins are going to be packaged into the different apparatuses of the cell, the endoplasmic reticulum, the Golgi, and they're going to be moved around by this microtubule system. That system isn't just doing it willy-nilly. There's actual processing, quantum level processing within those microtubule systems that guide what is going on in the cell. When different things come along, we'll say insulin again. When insulin is coming along, classic conventional theory is that it's binding to these receptors. Well, I go into very long detail in my book about this. It doesn't make a whole heck of a lot of sense. The amount of insulin we'd have to produce in our body to be interacting with every receptor of every cell in the body. I believe these insulin molecules and any other hormone, remember I was talking earlier about the aromatic amino acids and how those things are located within neurotransmitters, all the different communication molecules within the body, they have aromatic amino acids which are able to produce electromagnetic energy through those semiconductive nature, especially within those rings, the aromatic rings. So insulin is producing an electromagnetic message. These different receptors, they're acting as dipole antennas. They're not acting as lock and key mechanisms. They're acting as absorption of electromagnetic energy that goes into these protein networks. They eventually can go directly into these microtubule networks for processing. So that microtubule is going to find out there's a lot of sugar in the blood. Does the cell need sugar? If it does need sugar, it's going to uptake sugar into the cell. It can get so complex, we can talk for days on this. I leave it up to people that are in the profession to look at their areas of study. If you're studying testosterone, if you're studying cytokines, if you're studying any of the different chemical transmitters, if you look at it from a different perspective, that these are putting out biophotonic signals that go into this biophotonic network, this biophotonic processing system at the quantum level. That is how all interactions occur within our cells. So all cellular functions are occurring within these microtubule networks within a particular cell. There's interactions from outside the cell by other biophotonic messages. There's a master coherent signal that's going throughout the body that is received in a specific manner depending on the specific setup of the centriole. Like I said, I'm going to talk about that later, that the process of cellular differentiation is just a matter of hooking up these different dipoles and which groups are connected. Say we got one group of those, that three connected. This particular unit is going to have a different arrangement of what areas of the frequencies that they're working at. Final differentiation is just going to be a fine-tuning process. So a liver cell is going to have a very specific set of energies, of biofrequencies that are going to absorb into this system. Once that is interacting with this microtubule system, we're going to have genetic expression. We're going to have day-to-day function governed by that, different energy processing within it. The interesting part is, where is this coming from? That's going to be a whole topic. When I was talking earlier about that glial system, there's a master processing system that's coordinating the function of your liver cell, your kidney cell, your muscles of your big toe, your cones in your retina. All of these different cells are coordinating into one function based off of this particular frequency, that frequency that's going through those glial cells. So my next video is going to be on the glial network. I couldn't start talking about any of this until we discussed how this microtubule system functions, because the microtubule system of the glial cell network is going to be what's transmitting this information into the system, and then you're going to have a loop of communication between every cell in the body and the central network up in the glial cell network up in the central nervous system. So I look forward to doing that video. Again, I hope I didn't go through this video too fast. If I did, every topic that I discussed is detailed in my book, and it's got much better drawings and graphics than I produce on the board. But I feel that sometimes talking through something is easier than reading, so some of my readers may be confused. Hopefully my topics here and the way I'm discussing it helps. Thank you.